The immune response is initiated when a pathogen enters the body and thus some of its cells. Macrophages are white blood cells that encounter the pathogen, ingest and process the pathogen, then display its cell fragments on their cell surfaces, creating an antigen-presenting surface. White blood cells, called T helper cells, then recognize antigens and interact with the antigen-presenting macrophage. The macrophage and the T helper cells then release chemical signals that cause the accumulation of more white blood cells called cytotoxic T cells and B cells. Some normal cells of the body that get infected also digest the pathogen and present the antigens on their surfaces. Cytotoxic T cells accumulate that recognize many different pathogens. These cells then bind to the infected cells and kill it along with the pathogen. B cells also recognize specific antigens when they get activated by T helper cells to turn into plasma cells that are antibody factories that flood the blood stream with antibodies, which then bind to the antigens on the surfaces of infected cells and mark them for destruction. This is called the primary immune response. B cells can also become memory B cells, which keep the antigen memory of pathogens, so if the same pathogen enters the body again, the immune response is quicker and more efficient. This is called the secondary immune response and is how vaccinations work.